Welcome to this explainer. I am so incredibly glad you're joining us today because we are getting into something absolutely crucial: collecting, preserving, and submitting specimens for the laboratory diagnosis of bacterial infections. Now, you might think all the microbiology magic happens in the lab, right? Looking through microscopes, managing those high-tech incubators, but honestly, getting an accurate diagnosis hinges entirely on what goes down before that sample ever reaches the lab door. It is all about how you handle that specimen. So let's dive right into it.
So the crucial point we need to establish right away is a hard truth. A poorly collected specimen gives a poor diagnosis, regardless of laboratory expertise. In the tech world, they call this garbage in, garbage out. You could literally have a multi-million-dollar, state-of-the-art diagnostic lab, but is completely powerless if the sample they receive is already compromised. The lab relies 100% on your collection skills out in the field or the clinic. You are the very first, and honestly, the most important step in this whole diagnostic chain.
Let's take a quick look at our roadmap today for tracking a sample's journey. We're going to cover the high stakes of sampling, the rules of aseptic collection, matching your specimen to the infection, preservation and the cold chain, crucial submission protocols, and finally, biosafety and error prevention. Let's kick things off with Section 1, the high stakes of sampling. What actually happens when things go wrong right out of the gate?
Well, let's look at the two very different paths the sample can take. On one hand, a poorly collected sample just sets off this disastrous chain reaction. If you contaminate your sample with everyday environmental bacteria, the lab literally cannot find the real pathogen. That means false negative reports. And a false negative? That leads to incorrect therapy, prolonged illness, and, especially in veterinary or agricultural settings, massive unnecessary economic losses. But look at the alternative. If you nail a correct, sterile collection, you empower the lab to grow pure bacterial colonies. That directly translates to an accurate diagnosis, the right targeted treatment, and ultimately saved lives. The power really is right there in your hands.
Moving on to Section 2, Rules of Aseptic Collection. How do we get it right before it even leaves the patient?
All right, here are your 5 non-negotiables. Step 1, and this is massive. Always collect your specimens before antibiotic therapy starts. If you pump the patient full of antibiotics first, you're going to kill the exact bacteria the lab is trying to grow. Step 2, use sterile equipment. Absolutely no exceptions. Step 3, go straight for the active site of infection. Step 4, do everything you can to avoid normal flora contamination. Those harmless bacteria just hanging out on the skin or in the gut. And Step 5, ensure an adequate sample volume. Give the lab enough material to actually work with. Stepping out of this order severely compromises your entire mission.
Which brings us to Section 3, matching specimen to infection. Pathogens like to hide in different places.
Now, what's really fascinating here is just how diverse our aseptic strategies have to be based on the fluid or tissue we're after. Suspecting a blood infection, you're going to need a sterile venipuncture. For a respiratory issue where you need a nasal swab, that swab has to go into transport media instantly. Now, in the vet world, pulling a milk sample means you absolutely have to disinfect the teat first. Fecal samples? Got to avoid soil contamination because soil is packed with its own bacteria. Urine needs aseptic catheterization to bypass the lower urinary tract flora. And if you're dealing with pus or exudates, a needle aspiration with a sterile syringe is definitely your best bet. You really have to match your tool to the terrain.
I do want to quickly call out one very specific, crucial caveat when you're dealing with organ tissues, like during a necropsy. The tissue absolutely must remain moist, but whatever you do, never freeze it. It is so easy to accidentally ruin a beautiful tissue sample by just tossing it in the freezer. Freezing forms ice crystals, and those crystals basically act like tiny daggers that rupture and destroy the exact bacterial cells you're trying to preserve.
Let's jump into Section 4, Preservation and the Cold Chain, the perilous journey of the sample itself. During this transport phase, these bacteria need a lifeline. They're vulnerable and they are separated from their host. That lifeline comes in the form of transport media. These are special gels or liquids that keep the bacteria alive without letting them overgrow. Stuart's and Amy's media are super common, really versatile options. But take a look at Cary-Blair. That one is formulated specifically for fecal specimens. Picking the right transport medium is basically like putting the bacteria in the perfect little spacesuit so they survive the long trip to the lab. And the temperature for that trip? 4 degrees Celsius. That is your absolute magic number for short-term refrigeration and storage. It slows the bacteria's metabolism down just enough to keep them alive, but it stops them from aggressively overgrowing and totally skewing the lab results.
And again, repeating the warning from a minute ago. Unless the lab specifically tells you to for a highly specific test, avoid freezing at all costs.
We also have to be ready for specialized threats, like anaerobic organisms. These are bacteria, like Clostridium, that will literally die if they touch oxygen. They require strictly oxygen-free transport conditions to survive, period. If you grab a sample with suspected anaerobes and just chuck it into a standard oxygen-rich tube, yeah, that pathogen is dead on arrival, and the lab is going to find nothing.
Then we've got what microbiologists affectionately call the divas of the microbial world, fastidious bacteria. We're talking pathogens like Brucella and Campylobacter. Fastidious is just a fancy way of saying high maintenance. These guys demand super rapid transportation and absolute VIP temperature control. If they get a little too cold, a little too warm, or if the drive takes a few hours too long, they simply are not going to survive the trip.
All right, Section 5, Crucial Submission Protocols. The sample is finally arriving for check-in.
Just imagine being a lab tech and someone hands you a mysterious, completely unlabeled tube of fluid. It's absurd, right? You can't test what you can't identify. Every single specimen has to be fully labeled. We need the animal species and the owner's info. We need the exact date and time it was collected, because the lab has to know how old the sample is. You got to list the specimen type, your tentative diagnosis of what you think is wrong, and this is huge, any antibiotic history. If that animal's on penicillin, the lab absolutely needs to know that.
Listen, if you skip the paperwork, the diagnostic journey just ends right there. Unlabeled specimens, diagnostic labs will just reject them immediately. It halts the diagnosis completely. Furthermore, you must transport these specimens in leak-proof biohazard containers with ice packs to maintain that 4-degree cold chain. A leaking sample isn't just a ruined test, it is a legitimate, serious health hazard.
And this brilliantly illustrates how most diagnostic failures just come down to totally preventable mistakes. Things like collecting after starting antibiotics, letting a sample sit on a hot dashboard and delaying transport, or just using a non-sterile container. But when we flip that to correct practices, collecting before antibiotics, swift transport, proper temps, we guarantee the laboratory actually has exactly what they need to find the answer. Finally, Section 6, Biosafety and Error Prevention. Let's talk about keeping everyone safe.
Remember, we are often dealing with dangerous, sometimes zoonotic pathogens here, meaning diseases that can jump right from animals to humans. So laboratory safety isn't just about protecting the diagnostic quality of your sample, it's about protecting the actual people doing the work. You have to wear your appropriate PPE, gloves, lab coats, masks. Thoroughly disinfect those work surfaces, toss your biological waste into the proper safe bins, and always wash your hands after handling clinical specimens. Excellent science is always safe science.
Which leaves us with this final thought. You now know the rules of the road. Tracking a sample from an active infection site all the way to the lab bench. How strictly you follow those rules is entirely up to you. So, will your next sample yield a life-saving diagnosis, or just a contaminated culture? The foundation of an accurate diagnosis rests entirely in your hands. Thank you so much for hanging out with me for this explainer. Keep up the great work, stay curious, and I'll see you next time.